Hello everyone and welcome to another exciting webinar in our Biologics series. On behalf of DiscoverX, I would like to thank everyone for attending this webinar. I'm your moderator Poonam Kaul, who is the product manager for the GPCR Biologics and other cell-based assay services at DiscoverX. Before we begin this webinar, I would like to remind everyone that this webinar is designed to be interactive, and you can submit your questions by typing them in the Q&A box on your screen, and questions will be answered at the end of the talk. Today's webinar, titled How to Accelerate Your Biotherapeutic Development with Cell-Based Assay Services, is being presented by Dr. Neil Charter, who is the VP of Cell-Based Assay Services at DiscoverX. Dr. Neil Charter obtained his PhD from Imperial College London and worked as a scientist at UC Berkeley for 7 years before joining DiscoverX. He has been with the company since 2002 and has played an instrumental role in leading and developing the entire cell-based screening and profiling business within DiscoverX. In today's webinar, Dr. Charter will cover how DiscoverX cell-based assay services can enable screening and profiling biologics, with particular emphasis on our portfolio for immuno-oncology services. With this introduction, I would like to hand it over to Dr. Charter. Thank you, Dr. Charter.
Thank you, Poonam, and thank you for joining us today for this webinar. Today we are focusing on the use of DiscoverX's assays in the development of biologics for immuno-oncology and the cytokine space. DiscoverX's cell-based assays have been utilized at every level of drug discovery. Our cellular products and services fall in two major areas. Target-based cellular assays using proprietary EFC technology are particularly suited to screening and lead optimization. These assays cover almost every target class and are exceptionally strong in receptor biology. Our primary human cell-based phenotypic assays offer a powerful approach for understanding drug efficacy and safety in a range of cell-based systems that recapitulate a number of important disease models. Over the last 17 years, DiscoverX has built a large portfolio of assays that can be used to interrogate cellular interactions with particular focus on receptor signaling. This provides an exceptional depth of coverage across multiple target classes. DiscoverX's core technology can measure protein-protein interactions in recombinantly modified systems. This was first exploited to generate a range of GPCR assays that can detect second messenger signaling, beta-arrestin recruitment, and receptor internalization. The same approach has been expanded to examine tyrosine receptor activation and cellular signaling pathways. More recently, the technology has been adapted to measure receptor dimerization events and immune checkpoint regulation. Today's talk will focus on the utility of our checkpoint assays for developing the next generation of immuno-oncology therapeutics, as well as assays for interleukin receptor signaling that are relevant to immunological diseases. These have been effectively used by our clients to create bioassays for potency, quality, stability testing of biosimilars, and our services provide a cost-effective approach for characterization of biologics. These assays are particularly relevant for the development of novel biologic-based therapeutics, since they enable direct access or functional assessment of functional responses in critical new areas of discovery, such as immuno-oncology and cytokine signaling. This decade has seen the approval of an increasing number of therapeutics for the immuno-oncology leveraging checkpoint signaling. The majority of these drugs are therapeutic antibodies that combat the ability of tumor cells to evade the immune system. Biologics targeting the inhibitory checkpoint receptors PD-1 and CTLA-4 have been particularly successful and are being used for multiple indications. These have a proven track record in the clinic and offer an increasingly successful approach in cancer therapy. Data from clinical trials highlight how important these targets are in checkpoint therapeutic development. Over 80% of trials are currently focused on biologics modulating PD-1 signaling, either through blocking the receptor itself or more recently targeting tumor cells expressing the PD-1 ligand. CTLA-4 biologics make up the majority of the remaining trials involving checkpoint targets. Checkpoint receptors provide a unique opportunity for therapeutics that can target the ability of immune cells to evade the immune system. However, they do provide significant challenges when developing cell-based assays that can be used either in drug development, clinical trials, or production quality control. In particular, the assay often requires co-culture systems that involve the use of human primary cells. Protocols for assessing efficacy are often complex and have extended turnaround for completion. The assays themselves do not necessarily measure direct interactions for the target receptor, and as a result can lead to poor reproducibility or lack of specificity. PathHunter checkpoint assays offer a more direct approach by providing specific readouts on the key interactions that occur in co-inhibitory or co-stimulatory receptors. These model systems leverage native receptor biology to measure specific interactions with the target receptor. For co-inhibitory receptors, the corresponding ligand presenting cell can be targeted, providing the ability to develop anti-ligand or anti-receptor therapeutics. Since these assays interrogate native signaling systems using DiscoverX's well-established EFC technology, assay throughput is fast and data quality is highly reproducible. Checkpoint therapeutics address one of the key approaches that tumors use to evade the immune system. The checkpoint system exists to ensure that host cells are not targeted by the immune system. This is a highly regulated process and involves multiple components or checkpoints to avoid autoimmune responses. Two basic systems exist: co-stimulatory and co-inhibitory receptors. In the former, activation of the target receptor can induce immune responses. In the latter, interactions between the receptor and ligand-presenting cells serve to inhibit immune signaling and block escalation of the immune response. Tumor cells take advantage of this system by mimicking aspects of checkpoint signaling. For example, tumors expressing the PD-1 ligand can interact with immune cells expressing PD-1 to suppress the ability of the immune system to react. Therapeutics operate to block the ability of tumor cells to interact in this fashion. In the case of the co-inhibitory system, antibodies that bind to the receptor or the ligand on their respective cells prevent the inhibition of the immune response. This has been a particularly successful approach for therapeutics targeting PD-1, PD-L1, and CTLA-4. DiscoverX has developed a range of checkpoint assays that measure signaling for the majority of checkpoint targets. Co-stimulatory assays utilize endogenous signaling systems to measure activation of the receptors. Co-inhibitory assays employ co-culture systems to detect cell-to-cell interactions between ligand-presenting and the receptor cells. PD-1 signaling is a complex system with multiple interacting components. The PD-1 ligand is expressed on antigen-presenting cells that bind to the PD-1 receptor on T cells. This binding event triggers transmembrane signaling and phosphorylation of the inhibitory motif in the cytoplasmic tail by STAR kinases. SHP2 phosphatases are recruited to the phosphorylated tail, which in turn attenuate TCR and T-cell activation. The PD-1 signaling assay recapitulates these early events in this process by measuring the recruitment of SHP binding domains to the PD-1 receptor after phosphorylation. This is performed in a co-culture format where ligand and receptor are expressed on two cell types. Many variants of this assay are available, allowing the ability to measure binding of PD-1, PD-L1, or PD-L2 ligand-bearing cells, as well as the recruitment of SHP1 and SHP2 to the PD-1 receptor. One of DiscoverX's key technologies is the ability to measure protein interactions that are critical to many events in cell signaling. Our PathHunter assay technology is based on enzyme fragment complementation. This system is engineered from two fragments of beta-galactosidase. We call these ProLink and enzyme acceptor. Individually, these components have no enzyme activity, but together they recombine to form active enzyme that can hydrolyze the substrate and emit a luminescent signal which can be read on any standard luminometer. A key advantage of PathHunter assays is that cells can be engineered to express one or both of these fragments. These can be fused to proteins or localized to membranes or within cell compartments such as the nucleus. This enables the generation of assays that can detect protein interactions, protein translocations, receptor internalization, and modulation of protein levels. As noted earlier, there are multiple options for examining PD-1 receptor and ligand interactions using our assays. This system required the development of two cell types, ligand-presenting donor cells and receptor-expressing target cells. The two major species of PD-1 ligand, L1 and L2, exist, and the expression levels will vary depending on the tumor type. Ligand cell lines have been developed for both of these forms. These have been successfully used for identifying antibodies specific to one ligand over the other, resulting in the blocking of ligand signaling in co-cultured PD-1 cells. Both SHP1 and SHP2 PD-1 cell variants have been developed, allowing for the interrogation of specific signaling events using antibodies that bind to the receptor and block ligand binding. Whilst the primary use of these assays is for developing antibodies that block ligand or receptor binding, these assays can also be used to measure biologics that activate the receptor or even small molecules that block PD-1 inhibitory phosphorylation. The PD-1 co-culture signaling assays provide a robust signal to background. The assay window is dependent on the relative ratio of ligand-presenting and receptor cells, and a broad linear relationship is observed with the available ligand cells. Blocking of either ligand or receptor using specific antibodies reduces the bioavailability of the component to bind to its partner. Anti-human PD-1 antibodies disrupt signaling by binding to PD-1 and blocking the PD ligand. This approach yields similar results regardless of the ligand expressed. Comparable IC50s are obtained in PD-L1 and PD-L2 cells. The assay has a robust and sensitive response using commercially available anti-PD-1 antibodies. The assay is suitable for antibody therapeutics targeting either the ligand or receptor component. Here are examples of antibodies specific for PD-1 and PD-L1. The availability of assays specific to PD-L1 and PD-L2 ligands offer the ability to demonstrate and develop specific neutralizing antibodies to the respective ligand cell. PD-1 checkpoint assays have broad utility and produce potent responses for PD-1 specific immunotherapeutics such as Keytruda and Opdivo. The assays offer robust signal windows which make them ideal for the development of biosimilars targeting this important checkpoint. PD-1 is known to be phosphorylated with Src family kinases, which causes the recruitment of the SHP1 protein to the phosphorylated PD-1 receptor. Testing of PD-1 assays with small molecule Src inhibitors blocks the SHP1 recruitment to PD-1, demonstrating that the PathHunter PD-1 assay can be used to identify novel small molecule inhibitors for this exciting immunotherapy target. The assay is ideal for determining performance of anti-PD-1 therapeutics. Highly potent responses are observed for the PD-1 specific therapeutics Keytruda and Opdivo. Assays are also available for measuring responses for both SHP1 and SHP2 signaling, and comparable potencies are observed for Keytruda regardless of the SH2 domain. CTLA represents a second checkpoint target that has a successful result in the clinic. T lymphocytes can recognize and destroy cancer cells. However, an inhibitory mechanism exists in which activation of CTLA-4 interrupts this destruction. Ipilimumab is a monoclonal antibody marketed by BMS that turns off this inhibitory mechanism and allows lymphocytes to continue to target cancer cells. Cancer cells produce antigens which the immune system can use to identify them. These antigens are recognized by dendritic cells that present the antigens to cytotoxic T lymphocytes in the lymph nodes. The T cells recognize the cancer cells by those antigens and destroy them. However, along with the antigens, the dendritic cells also present an inhibitory signal. That signal binds to a receptor, CTLA-4, and turns off the cytotoxic reaction. This allows the cancer cells to survive. Ipilimumab binds to CTLA-4 and blocks the inhibitory signal, which allows the T cells to retarget the tumor cells. A 2014 study indicated that the antibody works by allowing patient T cells to target a greater variety of antigens. Checkpoint assays are highly specific as shown in these examples. For the CTLA-4 assay, the CD86 ligand cells produce a robust density-dependent response, whilst cells expressing PD-L1 are unable to induce signaling at any given density. This specific interaction can be blocked by anti-CTLA antibodies, such as ipilimumab. This first-in-class immunotherapeutic has been approved for metastatic melanoma. It has also been in trials for the treatment of non-small cell lung carcinoma and small cell lung cancer, bladder cancer, and some forms of prostate cancer. The selectivity of PD-1 and CTLA-4 assays is highlighted here. Commercially available anti-PD-1 and anti-CTLA-4 antibodies were tested in both systems. Potent responses were obtained for the selective antibody in each assay, whilst the corresponding reagent had no effect on ligand receptor signaling. This also highlights the high level of specificity that immunotherapeutics can impart. So to summarize, for co-inhibitory receptors, the DiscoverX checkpoint services offer unique features. The assays use stable cell lines that provide superior reproducibility over primary cell assays. The assay format produces greater sensitivity for bioactive molecules and is applicable for multiple applications, allowing for the interrogation of anti-ligand and anti-receptor biologics, as well as the development of small molecule modulators of signaling. We provide a fast turnaround that can fit the needs for lead optimization projects and can be customized to suit SAR programs. 5 stimulatory checkpoint molecules are the members of the TNF receptor superfamily. These are CD27, CD40, OX40, GITR, and CD137. Another 2 stimulatory checkpoint molecules, CD28 and ICOS, belong to the B7-CD28 superfamily. As the name suggests, co-stimulatory receptors are primarily involved in promoting T-cell expansion or the generation of T-cell memory. All have been subjects for drug development. AstraZeneca has 3 drugs in development targeting OX40. All of these are varying forms of OX40 agonists. ICOS, which is important in T-cell effector function, also has drugs in development. Johnson Therapeutics is developing an ICOS agonist. Co-stimulatory receptors can signal through canonical and non-canonical NF-kappa B pathways. In the latter, signaling is measured via the stabilization of the NIK protein. In the absence of stimulation, the NIK protein is kept at low levels in the cell through proteolytic degradation. Activation of the receptor blocks degradation, resulting in an increase in the abundance of NIK and subsequent IKK alpha mediated signaling. TWEAK is a multifunctional cytokine that controls many cellular activities, including proliferation, migration, differentiation, apoptosis, angiogenesis, and inflammation. The HVEM protein is a biomolecular switch that binds to the molecule light. It is both of these targets are members of the TNFR family and all signal through the NIK modulation. CD40 is a molecule found on a variety of immune systems, including antigen-presenting cells, and is transiently expressed on the surface of activated CD40 plus T cells. CD4 plus T cells. CD40 signaling is known to allow dendritic cells to mature and thereby trigger T cell activation and differentiation. The NIK system can be used to measure signaling via CD40, and the responses have been demonstrated using oligomerized forms of CD40L as well as agonistic anti-CD40 antibodies. Interaction of CD40L is complex, and soluble forms of the ligand generate muted downstream signaling. Oligomerization enhances both the CD40L efficacy and potency in NIK signaling. CD40 antibodies that have an agonist effect can induce the NIK pathway, and the assay is able to identify and rank antibodies that are bioactive for this target. This highlights the complexity of receptor signaling for and the importance of using functional assays to demonstrate efficacy in a cellular format. The OX40 receptor is a member of the TNFR superfamily of receptors, but is not constitutively expressed on resting naive T cells. It is a secondary co-stimulatory immune checkpoint molecule expressed after 24 to 72 hours following T cell activation, and its ligand OX40L is also not expressed on resting antigen-presenting cells, but is following their activation. OX40 and OX40L interactions have been found to play a central role in the development of multiple inflammatory and autoimmune diseases, making them attractive candidates for the intervention in the clinic. Conversely, stimulating OX40 has shown it to be a candidate for therapeutic immunization strategies for cancer and infectious disease. OX40 agonism promotes T-cell division and survival, resulting in the stimulation of both immune effector and memory functions, whilst also blocking the suppressive function of regulatory T-cells. This holds the potential to overcome immune resistance and enhance immune-mediated anti-tumor activity with OX40 agonism, particularly in combination with checkpoint inhibition. GSK have developed a humanized anti-OX40 agonistic molecule and is currently in phase 1 development. ICOS is a CD28 superfamily co-stimulatory molecule that is expressed on activated T cells. It is thought to be important for Th2 cells in particular. Ipilimumab patients express increased ICOS-positive T cells in tumors, tissues, and blood. The increase served as a pharmacodynamic biomarker of anti-CTLA-4 treatment. In wild-type mice, anti-CTLA-4 treatment resulted in tumor rejection in 80 to 90% of animals, whilst in gene-targeted mice that were deficient for either ICOS or its ligand, the efficacy was less than 50%. An agonistic stimulus for the ICOS pathway during anti-CTLA-4 therapy resulted in an increase in efficacy that was 4 to 5 times as large than that of control treatments. Therefore, combination therapy has potential to increase therapeutic outcomes, and the development of new biologics that can mediate different checkpoint targets will be important. There are currently 7 checkpoint assays available for testing using our service. 3 additional assays will be released shortly. Testing can be configured for a wide variety of applications depending on the assay type. Co-inhibitory checkpoint assays can be used in anti-ligand and anti-receptor formats and are also applicable for small molecule discovery or downstream shift signaling. Co-stimulatory assays can be tested for agonistic, anti-receptor antibodies, and other molecules that can modulate receptor activity. Interleukins are another area for successful biologic drug development. A number of drugs have been approved in the autoimmune area for rheumatoid arthritis and plaque psoriasis. Both therapeutic recombinant and monoclonal antibody approaches are relevant. Ustekinumab, or Stelara, is designed to interfere with the triggering of the body's inflammatory responses through the suppression of certain cytokines. Specifically, it blocks interleukin IL-12 and IL-23, which can help activate certain T cells. It binds to the P40 subunit of both IL-12 and IL-23, so they subsequently cannot bind to their receptors. Anakinra is a modified version of the naturally expressed IL-2 receptor antagonist that blocks the biological activity of IL-1. IL-1 is involved in inflammation and cartilage degradation associated with rheumatoid arthritis. The antagonist operates by competitively inhibiting the binding of IL-1 to the interleukin-1 type receptor. Tocilizumab, or Actemra, binds soluble as well as membrane-bound interleukin-6 receptors, hindering IL-6 from exerting its pro-inflammatory effects. Canakinumab, or Ilaris, is a human monoclonal antibody targeted at IL-1 beta. It is highly specific and has no cross-reactivity with other members of the interleukin-1 family. A broad range of interleukin receptors are available for screening at Discoverex. These assays are ideal for the development of biologics since they directly measure interleukin interactions. The initial event in interleukin signaling is dimerization of receptor subunits upon ligand binding. PathHunter interleukin assays measure ligand-induced dimerization by tagging receptor subunits with the two components of the enzyme fragment complementation system. As the two subunits are brought together by ligand binding, EFC enzyme activity is formed. Since a stoichiometric relationship exists between ligand binding and enzymatic readout, the assay is ideal for measuring both ligand activation and inhibition. Interleukin specificity is conferred by heterodimerization of different receptor subunits. For example, IL-12 receptor can form homodimers or heterodimers with IL-23 receptors. IL-12 and IL-23 sequences are highly conserved and differ primarily through the N-terminal part of the proteins. PathHunter interleukin assays can distinguish these ligands by selective tagging of the receptors of interest. As a result, these assays are highly specific for their respective ligands. This is particularly important for the development of antibodies that can selectively bind specific receptors or ligands. Over 30 assays are available for interleukin discovery and are applicable for multiple applications, including activation, receptor blocking, ligand neutralization, as well as neutralizing antibody assay development. So in conclusion, Discoverex's checkpoint and interleukin services offer a cost-effective and efficient means to screen biologics for these important target classes. These provide highly specific results that are based on native receptor biology. The assays are ideal for the development of immunotherapeutics and can be used in a wide variety of applications. Our fast turnaround provides the means to quickly test molecules to rapidly drive your drug discovery program forward. Thank you for taking the time today to attend this webinar. Please post questions in the Q&A session, and I will try and answer as many as possible.
Thank you, Dr. Chata, for the very nice presentation. I would like to remind the participants that they could post their questions on the Q&A box or the chat box. Also, we will be sending the webinar slides to the participants after the session. So here, before we begin, Dr. Chata, I would like to ask one question. When do you recommend using the SHIP1 versus the SHIP2 recruitment assay?
Yeah, that's a good question. A lot of papers have focused on the SHIP2 recruitment assay. This seems to be the primary SH2 domain protein that is recruited to the receptor upon phosphorylation. However, there may be instances where SHIP1 becomes a relevant target.
Okay, very good. There's one question coming in. Can checkpoint assays be used for biologics or small molecule development?
Yes, for sure. The primary use for these assays has been in the development of antibody therapeutics, but it's also possible to look at the downstream signaling that occurs with, especially with the with both types of checkpoint assays, to look at molecules that can actually interfere or modulate the signaling with these assays.
How does your anti-ligand format vary from the anti-receptor format?
Yeah, that's a great question. So if you're looking for molecules, or especially antibodies, that are going to bind the ligand on the antigen presenting cells, then you actually want to incubate the antibody with those cells prior to adding them in to the receptor cells. Likewise, if you're looking to measure antibodies that can block the receptor itself, then you will incubate the antibodies with the receptor cells first before adding the ligand cells.
There's another interesting question that has come in. Can you detect agonists in both the CTLA-4 and PD-1 signaling assays?
Yes, for sure. Those type of molecules will primarily act on will act on the actual receptor cells. So increasing or modulating the activity or activating the receptors on those cells. So the format will be slightly different, but we certainly have seen interest in that for certain therapeutic applications.
So there's another question. What does your interleukin assays measure?
So the interleukin assays measure the dimerization event that occurs when a interleukin actually binds to the receptor. Since these are highly specific assays, we actually build different cell lines that have the respective subunits that are required for that particular interleukin assay. So once the ligand binds to the receptor pair, it causes dimerization, and then we can actually measure that event using our ESC technology.
There's another question coming in. Have you tested soluble ligand in the PD-1, PD-L1, or CTLA-4, CD86 assays?
We have looked into that. There are certain molecules that may modulate PD-1 or CTLA-4 activity, and they can either block or activate the system. And these can be very interesting in that they're more open, they're easier to develop novel drugs, or they can actually be derived from things like microbiome or different types of approaches for looking at novel chemistry.
Very good. So one last question, Dr. Chatter, what is the typical turnaround time for your services?
So our average or standard turnaround time for services is 15 days. If you need an accelerated turnaround, we can do 10-day turnaround, and for SAR programs, we can do as quick as 5-day turnaround.
Okay, very good. Looks like we don't have any more questions. Thank you, everyone, for attending the webinar. I would like to remind everyone that we will be sending a presentation after this session. If you have any questions, you can email them to pcall@discoverx.com, or if you have a tech question, you can reach out to our technical service team, which is also indicated on the slide deck. Thank you, everyone, and have a good day. Thanks, Dr. Chatter, again. Bye.
Thank you very much.